they have been co-evolved with bats for millions of years, and they don't cause disease in bats. All right. So here are some bat facts. Although I did my undergraduate degree in biology in Huadong Sida East China Normal University, but I'm really an engineer-minded person, so that's why I chose biochemistry and molecular biology. So I don't have a native sort of affection for animals. So bat research is purely because of the viruses I have been studying. Then eventually, of course, we try to know a little bit about bats. So bats are very successful mammal. With the living mammal species on Earth, currently we have over 5,000, 20% of them is a bat. Only in terms of species diversity, only second to rodents. And that they are the only flying mammal on Earth, and they're more widely distributed than any other mammal except human. Nowadays we have airplane and ships, we can go anywhere. And they have variable thermoregulation, echolocation. But the last 3 points I want to emphasize is that, I don't know how many of you know that bats has the longest lifespan, the relative lifespan, right? For mammals, your longevity lifespan is proportional to body size, in proportion to your metabolic rate. And bats defy both of these rules. They're small and they have a high metabolic rate. And I know you have a lot of cancer biologists in this audience. Bats is also a great model system, but because they seldomly develop cancer. And then for us, of course, I lead this, pioneer this field to discover that bats carries a lot of viruses. Even more interesting is during flight. All bats fly, right? And their metabolic rate can go surge to 30-fold of their base rate. Their heartbeat can get to close or over 1,000 beats per minute, and their body temperature can go to all the way to 42 degrees C. And I always say, you can imagine any of you in the audience right now, if you do this, you will not last an hour, you would die basically. But for bats, that's normal physiology. They do that every night. Even more amazing is like the bats in your part of the world, in North America, these bats go through hibernation in winter. So during hibernation, the same bats, the heartbeat can go to 6 beats per minute. So they beat once and the rest for 10 seconds. So for the cardiologist in the audience, I already have a collaboration with the Imperial College of London. We're studying the bat heart. It's incredible, so there's lots to learn from bats. So I was the first one, to really dig into the bat genome and try to understand what makes bats so special. Of course, we went in to ask why there's such a good reservoir, for viruses. So this was published in 2013, and the flying fox from Australia was used as cover. What we discover, of course, there are many things we can discover, but the 2 most interesting, really high impact discovery is that compared with the land mammals like human and mouse or any other mammals, the most interesting difference are 2 pathways. One has to do with DNA damage repair. So bats have a much more efficient and really positive selected genes in that pathway. On the other hand, bats try to do everything to reduce overinflammation. I'm sure, in the audience, many of you have medical or doctors or medical scientists. Inflammation is such a hot area. Inflammation is involved in almost all human diseases. So bats are healthy, we think, is because they can control inflammation, just get the right inflammation level. We're not alone. So we published that paper in 2013, and 5 years later, this Cell paper, led by Gustavo from NIH, and they really sequenced the bat genome for another Egyptian Rosetta's bat. That's the bat that interested because it's a reservoir species of Ebola virus. Without going too much detail, focus on here, left bottom. Basically, in terms of infection of virus in bats and a human, they can see that the antiviral effect is similar. So bats also can control the virus can replicate in bats. The difference is that the inflammation. When we get a viral infection, we go crazy. Our inflammation system is turned to very high, sometimes over. Whereas bats have inflammation, but it's very controlled. So the same day this paper come out, there was a commentary, in the Cell press in immunity. So immunity, and they have this paper, review paper with a very catchy title called "The Holy Immune Tolerance Batman." So they try to emphasize to say, you know, from this paper, the key discovery is tolerance. They don't inflame much. But to me, this is a one side of story. I will tell you later that it's not the wholly immune tolerance that's different, you know, from bats to human. It's the balance, the wholly immune balance. Because in my group, in the last 7 years, we have published many papers to demonstrate that the bats actually have elevated the innate immunity, but also have the dampened innate immunity that depends on different pathways. So, you know, I mean, in Chinese medicine, that is all about yin and yang, right? How to keep you healthy is to get the balance right. But I think in Western medicine, we now more and more believe it's the same principle. To keep healthy, you have to get the balance and a defense and tolerance right. Not enough defense, then the cancer cells and the virus will replicate like crazy and it can eventually kill you. If you have too much defense, then sometimes you get overboard and you have, you know, inflammation disease and autoimmune disease. But whether you're a bat or human, you know, evolutionarily we have this selection and we evolve different ways. So there are differences between species, right? But also individual level, we cannot get our biological system at a balance all the time. Then I think it becomes a computer, right? A machine, you know. As a biological individual, that you change it, your emotion changes, your sleep, jet lag, you know, food you eat, everything. And so we are constantly adjust on a daily, hourly basis to try to get to the balance right. The difference is that for humans, that range of adjustment is greater than if you're a bat. Bat has a much narrower range they can control. That's why they have a better balance. Of course, you know, this is out of all real data and I don't have time to go into this. So just to give you a very simple diagram, these are defense genes, you know, so which are interferon genes, heat shock protein genes, you know, DNA damage, and so on. So as you can see on the right, we're very, very familiar with this kind of scenario, right? So these are all in cell culture, not in our body, so in vitro. So if you give enough nutrient and 37 degrees, the cells, human cells happy, they don't switch on defense genes. But if you induce them, so this inducer 1 and inducer 2, induce 1 is chemicals. If you put on chemicals, they go up. And sometimes we also use virus infection, so they go up. As you can see, these are log scale, right? From base level to all the way, you can be 5, 6, 7 logs. For bats, it's different. Some of the genes already switch on before you see enemies without danger signal. And when you put a chemical or virus infection, they go up, but not by much. So the range is narrow. So this is the basis of the animation I have in the front. Currently, we are doing a review for Nature. So this is one of the figure. Basically, we are saying that, you know, we just give examples, you know, for example, the interferon genes, the heat shock protein genes, the autophagy, and the efflux pump like the ABCB1 for cancer biologists, you may know this. These proteins or genes are, the basal level is much higher in bats than humans. On the other hand, the STING molecules, I1 beta, the multi-gene family in the pi gene, the inflammasome sensor, the NAP3, all these are dampened. Okay, so they different from us. So I just want to, you know, today I'm not going to show you much more detail than that. Really, it's the conceptual seminar. And what most really fascinating is the inflammation control. So we have published a few papers, and one just accepted in PNAs. It'll be coming out hopefully in a week or two. So this is the human mouse. Okay, on the right side is the bat. So inflammation is so important in our body. So there's many levels of control. So the inflammation, first you have a priming that, you know, comes in, and then you have the activation. Activation, you have a cascade of molecules involved. You know, it goes from a sensor, it goes from ASC, you know, then a caspase, and then you come to I1 beta, and I1 beta triggers downstream, you know, defense. We have discovered 1, 2, 3, 4, 5, 6 different mechanisms in bat, you know, in the last 7 years. All of them are trying to regulate that process so that it does not go overboard. So inflammation dampening is absolutely very important. And I just show one result we published last year in Nature Microbiology is really in the context of why we got sick when we get COVID-19 or SARS or MERS. and bats just totally healthy. So we did that experiment using most virus, okay? So just to go through this, is human PBMC, mainly some of the immune cells, and we have bat PBMC. So in a biocontainment facility, BSS3, we can infect these fresh blood cells from human and bat by MERS coronavirus, the live virus. And then we scan for the spike protein to see the virus replication. So the green color means the virus is replicated. As I said, the antiviral pathways of human and bats are not much different because the virus still can replicate to a very decent level. The middle is, this is an image string. So this is our ASK1 molecule. So once you get a viral infection, if you activate the inflammasome pathways, these molecules will come together, aggregate, and form a very compact, type dot in the image string. So we can see that. So this red dot means it's fully activated. And you look at it here, nothing. We cannot see anything. We know it's activated, but less than 10% of human level. That's why you cannot see on this slide. So basically, this is a very typical example of, to say, a pathogen comes to human and the bats, they get infected, they replicate, we have immune response, but the response is very different. And unfortunately, it's that immune response, especially the inflammation, that if you go overboard, then you cause severe disease and death. So in our field, we have a cliche, we say very few virus kills us, we kill ourselves. So just to summarize that part quickly is that bat has a much better host defense tolerance balance. And of course, that did not come overnight. That's 65 million years of evolution, because the flight capability gives bats advantage, but also disadvantage. So they have evolved these 65 million years, become the modern bats. Now the modern bats have a slower aging process and a long lifespan, less protocancer, less inflammatory disease, less autoimmune, less metabolic disease, less everything. And we just got another 3 grants to study this. So as I said, we always say, it's not bats' fault we have COVID-19 or SARS. It's humans' fault, really. Bats have been on Earth for much longer. But today, what I try to say is that, this is also the fountain of new knowledge. So if we dig into bat biology, unfortunately, yes, bats give us a virus, but what I try to say is that, bats is also going to be our teachers. We can learn a lot from bats to benefit human, and also to really facilitate our human medical and health research. So now come back to these slides. I started to say all these emerging zoonotic viruses of bat origin in the last 25 years. And I was involved in all of them, but more heavily in the first 3 or 4. All of them, serology play a very important role either to identify the origin or identify the intermediate host, okay? So I was in Wuhan, January 14th to 18th. So I was talking to the scientists on the ground with Wuhan Institute of Virology. Before I came back, I already ring my postdoc to say all the genes I want to do serology, because I already envisaged that COVID-19 is much bigger, more serious than any of the previous virus I have studied, and it's going to stay. So we have challenges. The challenges for COVID-19 serology, for other viruses, like a SARS, eventually we got to eradicate, but for COVID-19, it's going to stay, right? So what we have is these challenges, in red, these are the 5 dot points for human. Currently, we're talking about exit strategy, and you guys in California are much in a worse situation than us in Singapore. But even in Singapore, we're worried about herd immunity. How can we allow people to go back to the concept of immune passports? Even if you're infected or vaccinated, how long the protect immunity will last? And can we use convalescent plasma from a survived patient? Do they have enough neutralizing antibody for therapy? I'm pretty confident we will have a vaccine, more than one eventually, but how do we know the vaccine is efficacious, not only at the population level, but you as an individual? If you go get that vaccine shot, 1 or 2 shot, how do you know you actually have the correct and high appropriate level of immune response? And the last is my area, right? We still don't know exactly which virus and how they got to Wuhan. So Serology will play an important role in all of this, but unfortunately, the serology kits now, more than 100 on the market, none of them can really do this job, because what we need is we need to measure the functional antibody, which is the neutralizing antibody. So for the human application, we need a neutralizing antibody, and for the animal, we need an antibody test, which is species independent, right? Because we don't know which animals, you know. When I was search for the origin of the hantavirus in Australia, we tested 42 different animals, including koalas and kangaroos. But we are not going to be able to develop a specific serology for each animal. So I want a species-independent serology test. Here comes our invention. So we invented something called a surrogate virus neutralization test, SVNT. Basically, it addresses both challenges by determining neutralizing antibody in a species-independent manner. So I'm going to talk about the scientific part, and the next talk by Dr. Zhu will talk about the commercial development. The paper is already out in Nature Biotech, so I'm not going to go too much detail. Really, for this audience, I try to really, you know, educate you, try to understand how important, how it works, right? So most of you now have seen this image many, many times on TV. So this is the coronavirus, the red triangular things, you know, protruding out of the surface. If you do a section, these are the spike protein, right? So this is the genome of coronavirus, you know, 30,000 nucleotide. And so what you have is, of course, there are lots of proteins encoded. When a virus like that size infects you, your body is going to launch immune response and produce both T cell and B cell. From the B cell, you get lots of antibody, hundreds, if not thousands. But all of them almost are binding antibodies. So they basically bind to a specific protein and a specific region we call epitopes. Among the binding antibodies, there is a very small proportion. For coronavirus, that's at a 1 to 3%. That's the real important one, the neutralizing antibody. And we know that this spike protein is the major target for neutralizing antibodies. So this is the green protein in gene here. And with the spike protein has more than 1,000 amino acids, only one region, around 250 amino acids called the receptor binding domain, that's the most relevant for neutralization. So this is, you know, traditionally, virus neutralization test is the gold standard for serology antibody. How that work? You have a virus comes in, you bind to the cell surface. In this case, for COVID-19, you bind ACE2 and get the infection and you kill the cell. If you have neutralizing antibodies, you can bind to the virus, especially that IBD region, and you block that process, and then the cell is healthy. Its gold standard is in specificity, but in sensitivity, actually, it's not very sensitive. You know, you have other assays. But the disadvantage is, first of all, you need a BSS3 lab, you know, and you need to protect yourself like that before you can do anything. Very tedious and slow, takes 3 to 5 days. You need a highly skilled staff because that's the readout, you know, you cannot use the machine. Very expensive. You know, if I charge you a real cost, the test will cost 2,000 to 3,000 US, one test. So our invention is very simple. You know, as remember, I was trained as a biochemist. So we did a biochemical simulation, basically. So this is a diagram. You have the receptor ACE2, which is a membrane protein. You have the spike protein, which is the membrane of viral protein. And when they interact, then you get an infection. And if you're neutralizing antibody, you block that, you neutralize the virus. So we engineer the protein with the help of GeneScript. We express the protein as a soluble ACE2, so we can code directly onto ELISA plate. And for the spike protein, we only express this 250 amino acid receptor binding domain. To make the life even easier, we chemically conjugate this with host register peroxidase, the most commonly used enzyme for ELISA. So we turn a virus, live virus BSS3 5-day experiment into a simple ELISA on the plate, which can be done in 1 hour in any open lab, including hospital diagnostic labs. So the principle is very simple, right? This IBD binds with ACE2. If you have the neutralizing antibodies, then this is reduced and you see a reduction. So these are very simple data. We express the IBD, the spike protein, and the nucleic acid protein, which is the internal protein, should not interact with ACE2 as a net control. And you see a dose-dependent binding. So that's brilliant. IBD is the best. In the presence of human sera and the net control sera, you can see the COVID-19 patient sera have a dose-dependent inhibition. The negative control sera does nothing. And the other beauty is that, COVID-19 and the SARS are so close. They both use the ACE2. So in my lab, of course, I have been studying this for the 17 years. So immediately when we get this data, we, of course, went to a patent application, but also we transfer this to SARS. Now we have a SARS coronavirus IBD, SARS patient sera, and SARS negative sera, and exactly same we achieve. So now we have surrogate virus neutralization for COVID-19, for SARS, but I don't have time to go in. Now we have established this for pangolins, for bat viruses. So the research, we can do much, much more detailed fine-tuning of virus neutralization now. So just to validate in patients, these are PCR positive patients, in Singapore and in Nanjing, in China, as you can see, that the positive and the negative, we have really good specificity and sensitivity. And we can correlate basically this surrogate virus neutralization versus conventional virus neutralization. Means the conventional virus neutralization, you have to use the live virus, goes inside of BSS3, but the correlation in R square is close to 0.9. And beautifully, we can differentiate truly that binding antibody versus neutralizing antibody. So these are monoclonal antibodies, 4 of them selected by binding to the IBD. By the way, this is exactly the same molecule used in ELISA and in our surrogate virus neutralization. So the consideration of post-translation modification, conformation, everything is identical. So you cannot argue about that. So we did a direct binding in ELISA, all 4 monoclonal, especially that red one is the strongest. And then you change the format, you put in the ACE2 and ask for inhibition or neutralization. Now, only 2 of them are neutralization positive. The other 2 is negative, including the strongest one, the red one. So this is what we try to convince FDA to say, this is the only truly neutralizing antibody assay that you can do outside a biocontainment facility. Just to finish up is this species-independent neutralization. This is close to my heart because I really want to use this to search for the origin and the intermediate host. So this is a COVID-19. So we have the rabbit sera and the mouse sera. As you can see, beautiful inhibition, and the negative control has nothing. This is the SARS coronavirus. We have rabbit and also immunized sera, but we also have civis, ferrets that I infected when I was in Australia. And this is a ferret sera, as you can see, also inhibit beautifully. So, and Malik Paris is a very big name in our field from Hong Kong University, who was involved in SARS, MERS, COVID-19, So Malik has been adopt our surrogate virus neutralization very early on. So this is a preprint, hopefully it will be published soon. He has used this for human, dogs, cat, hamster, and other groups have used, So Sumi introduced this zoonotic transmission concept. It's an animal to human transmission, but it's actually, zoonosis just means that between different animal species, and the human are considered animal as well. So to differentiate these, we have a terminology called a spillover, means animal to human, and spillback is human to animal, or forward zoonotic transmission or reverse zoonotic transmission. Forward is animal to human, and reverse is human to animal. In all the emerging zoonotic disease, I have described 6 of them. We have never been worried about the reverse transmission because most of the, like SARS, Hendra Nipah, eradicate. For Ebola, it's very limited in geographic location. But COVID-19 has changed all of these. The reverse zoonotic transmission is not a theoretical risk, it's real. I mean, you guys must heard a lot about mink farms now, not only in Europe, but in USA, infected, and this animal can actually die and get disease from. So in addition to mink, now we have evidence human can pass COVID-19 virus to cats, dogs, and tigers. And I'm sure many more is gonna come because all of us, humans and animals share this ACE2 receptor. The ACE2 is highly conserved, and many animals have the ACE2 that the virus can use and enter. So in terms of animals in emerging zoonotic virus, up until this year, I have been working this for 25 years. we have been focused on this as a natural reservoir, amplified host, or intermediate host. But now COVID-19 have opened up this two new. One is a spearback host, and the other is what I call unnatural natural reservoir. So what I mean is in these slides, right? My current working hypothesis is the virus come from bats and most likely in Asia, not necessarily in China. I will come back later. Goes through an animal X. Currently, we think it's pangolin or something else. It's a mammal, most likely. Get into human, massive human-to-human transmission, and spread to the world. And then, if you have a close contact, you can get into pets and farm animals like minks. Mink can pass this animal back to human, and that's already demonstrated. So this is kind of scary already, but it's more scary is that scenario. For you guys, where you stay in North America, South America, you have bats, and we know some of those bats are susceptible. Imagine human pass the virus to bats, bats will not develop disease, right? So you will not know. And then the virus will stay happily in this unnatural natural reservoir, replicate, mutate, and they will spill over into other animals and then to human, okay? So that cycle, once that established, it's very, very hard to eradicate that, and this virus can travel around the world like crazy. So this is a commentary just come out, the Economist, and by two leading scientists, so I work with both very closely. Peter Daszak is the head of the EcoHealth Alliance. If you are paying attention, he get very famous because his grant, NIH grant, was cut by Trump after his White House press conference. And that's the NIH grant that I'm a co-PI as well. So Peter Daszak and I have been collaborating a long time. Jeremy Fry is the CEO of Wellcome Trust. Both are well-known infectious disease experts. Both are very, very adamant to say that we need to expand this study into Southeast Asia if you really want to find the origin of the virus. The reason is that this is the diversity of bat species, especially the rhinoceros we know carries SARS-CoV-19 virus. Susan Lee's group from Wuhan Institute of Virology discovered that RATG13 virus from a cave in Yunnan province 7 years ago. Okay, so in China, the bat diversity goes more diverse when you travel south, but nothing compared with Myanmar, Laos, Vietnam, Thailand, Malaysia here, and Indonesia. So this is the area, really is the playground, for bats and viruses, okay? So I think, I have started this work already in this area, and our SVNT will play a very important role to get to this. So I will finish up with these lessons we learned from emerging zoonotic virus. I think, this is from Hangzhou to Wuhan. So 25 years, I'm not going to really, the paper is published, you can read about. I just emphasize this, like in China, there's a lot of debate about outbreak response versus scientific publication. But what's not in this commentary is the other lessons. The double edges of social media. I mean, COVID-19 is really incredible, It's a waking up for us. And also, I have to emphasize, it's not bats' fault. Bats has been around for a long, long time. The last, without being political, but I think it's really, I have been working in this field for 25 years. We play a very important role for SARS. And in those days, we all fight pandemic without politics, but now we have politics. And to me, mixed pandemic with politics is a big disaster. And I just want to finish up to illustrate a point. In this, the joint mission, I attended 2003, WHO, FAO, and the Chinese government, 8 scientists from the international community. And, we have 2 Australians, 2 French, Japanese, Americans, Filipino, and from Hong Kong. Especially this American scientist, Ann Shuka. She, at that time, was a division chief, and now is the deputy director of CDC. And this is, she is in White House with the press conference. So imagine that, if we assemble a team to go to China to study the origin of COVID-19, that's not going to happen, right? Right now, Australians and Americans cannot play that important role. Same, in 15 months later, after this WHO mission, I was able to assemble a team of scientists from China, USA, Australia. We work as one. Right now, I don't think that's possible. So that's a sad story. But I will finish up with thanking my backpack team in Singapore. And these cute guys, they changed my life, you know, in terms of my bat virus and the bat biology, bat immunology studies. And I thank you for your attention.